In this video, we're gonna continue on introduction to parasitology. In the previous video, we talked about the terminologies used in parasitology, and we're able to define parasites as organisms that lives in or outside the host, derives food, shelter, and nutrients from their host, thereby causing harm to the host. And we classify organisms, we classify parasites into different classes according to their habitat, according to their dependence on the host, and other ones like that. And we talk about vectors, which are living organisms, usually arthropods, that transfer parasites from one host to another, usually from an infected host to a non-infected host. And these parasites can be further classified into two, which is mechanical parasites and biological parasites. We have talked about that. So in this video, we talk about infection. And what is an infection? An infection can be defined as the process of entry and subsequent establishment of the pathogens and resultant damage to the host. Are you getting that? That is what an infection is. Now, we now have different stage of an infection. We have the diagnostic stage. The diagnostic stage is the developmental stage of a pathogenic organisms that can actually be detected inside stool, blood, urine, sputum, and CSF. CSF means cerebrospinal fluid, and other human body secretion, and that is the diagnostic stage. The infective stage, that is the stage of the parasites that is capable of entering the host and continue development within the host. That is the what? That is the infective stage. I've talked about infection. I said infection is the process of entry and subsequent establishment of pathogens and resultant damage to the host. That is what an infection is. Now we now have zoonosis. Zoonosis can be defined as the disease of animals. Disease of animals that is actually transmitted to man. Example of that is Lassa fever is a zoonosis because it transfer from, I think, bats. Rabies is a zoonosis transmitted from dog and other ones like that. So that is zoonosis. We now have incubation period. We have what? We have incubation period. And incubation period is the time between the initial infection between the initial infection with the parasite and the demonstration of the parasites or their stage in excreta, blood, and other diagnostic material. Which means what this one means is that, for example, you are bitten by malaria today, but it is not today that you are going to start having the malaria. The malaria will undergo some processes. When you start feeling the symptoms, the time at which the plasmodium enters your body to the time that you start feeling the symptoms of malaria, that is incubation period. Are you getting me now? Or I can define incubation period in my own word as the time between inoculation of pathogens to the time of appearance of typical symptom. That is incubation period. Are you getting me now? So that is another definition in my own side. Now we now have some other things like super infection, retro infection, and other things like that. So those ones are like that. Now moving from there to source of parasite exposure, source of how you can be exposed to parasites. How you can be what? How you can be exposed to parasites. The first one is through contaminated soil. You can be exposed to parasites through contaminated soil or polluted soil with human excreta. And example of this is an example of parasite that you can actually contact during exposure to contaminated soil is Ascaris lumbricoides, Stecholaris, Trichuris trichura, and hookworm is an example of parasite uh, that you can actually contaminate during the what? During contamination, um, contaminated soil. Now, the second one is contaminated water. So another example is how you can be exposed to parasitic infection is contaminated water. A water may contain a viable cyst of amoeba or Taenia solium eggs. Can also contain the sarcaria of a blood fluke, that is Fasciola gigantica. Can contain a cyclus of Dracula mendinensis. Can contain um, Fasciolopsis busci. Can contain lung fluke and different parasites that you can actually contain from what? That can actually enters through your body through contaminated water. The third one is insufficiently cooked meat or pork or beef. An example of that is Taenia species, that is Taenia solium and Taenia saginata. And it can also be contacted through blood-sucking arthropods like malaria parasite, it is caused by Anopheles mosquito, leishmania, trypanosoma caused by Glossina species, and Wuchereria bancrofti um, caused by mosquito. And the mosquito is called um is called Kuline mosquito. Is the one that causes Wuchereria bancrofti. Is the one that carries Wuchereria bancrofti. Now, moving from there to the next one, can also be contacted through animals, like I said, like zoonosis, dogs, herbivores, and other animals like that. You can be exposed to parasitic infection through them. Also, human beings. Human beings is responsible for contamination of parasites, like through kissing. You can contaminate a particular parasite known as Trichomonas tenax or Entamoeba histolytica or Entamoeba vermicularis and other ones like that you can actually contact from them. Now, through sexual intercourse like Trichomonas vaginalis can be contacted through sexual intercourse. So the last one is auto-infection. Auto-infection is like you have it right from birth. Are you getting me now? Right from birth, like Taenia solium. It is not a must for you to eat cow or pork or beef because before you can actually contact Taenia solium. You understand? Before you can be exposed to Taenia solium. I get me. That one is auto-infection. Now, moving from there to a mode of transmission. We have two mode of transmission. We have the direct mode of transmission, and this one it requires a biological vector. It does not require rather does not require a biological vector. That is direct mode of transmission. For example, Ascaris lumbricoides can actually enter through into a human by eating of sand. You are the one that go to the sand and eat it yourself. No one pushed you there. You understand? So if you take in the egg of Ascaris lumbricoides, then no vector transmit it to you. You are the one that took it. Or taking of contaminated water. You understand? Or penetration of the skin going to river. You can like the cyclops of Dracula mendinensis can actually enter into the body. You understand? Through what? Through river. That is direct penetration of the what of the skin through insect bites. I said it already. And now the second one is indirect contamination. I get me now. The second one is what? Indirect contamination. And the indirect contamination is the one that requires a biological vector for the what? For the transmission. I get me now. The indirect transmission. Now, we now have effect of parasites on their hosts. What are the effects of parasites on their host? The first one is consumption of nutritive elements. That's the first one. The consumption of nutritive elements. That is they absorb nutrients from their what? From their hosts like hookworm, they suck blood. The second one is obstruction of passage. When they are much, like Ascaris lumbricoides. Ascaris lumbricoides, when it is much, it can cause obstruction in the intestine, whereby food won't be able to what? Food won't be able to pass into the what? Into the small intestine. I get me now. They can cause bleeding like cystosomia hematobium when someone stool, when someone poo poo, it is going to come out with blood. You understand? These are it can cause bleeding. Destruction of tissues, they can destroy tissue, they can cause necrosis in liver. You understand? Compression of organs, they can compress organs and they can release toxic substances. The rupture of granulosous cyst, and even the normal Plasmodium parasites, they release a toxic substance called emozoin. And emozoin is a toxic substance that actually, when they infest on the hemoglobin, they release this particular substance. You understand? The seventh one is opening pathway for secondary infection, like ulcer. Ulcer can be formed as a result of Dracula medinensis, which leads to exposure to bacterial infection. You understand? That is, the open pathway to secondary infection. Allergy development, like you can be allergic before because of parasites. You can actually be allergic to something that you are not allergic to before. Transmission of pathogens to man, and they can cause chronic immunologically disease. These are effects of parasites on their host. Now, moving from there to the last part, the life cycle. What is a life cycle? We can say a life cycle is just it's just the stage of life of a parasite from the larval stage to the adult stage, back to the larval stage, back to the adult stage. You understand? That is what a life cycle is. We now have two types of life cycle. We have the direct life cycle and the indirect life cycle. The direct life cycle is also known as monoxenous life cycle. Alright, monoxenous, monoxenous, monoxenous life cycle. And the indirect life cycle is also known as heteroxenous life cycle. Hetero, heteroxenous life cycle. So these are the two type of life cycle that we have. And the direct life cycle is also the direct life cycle, also known as monoxenous life cycle, is a development process that does not require any intermediate host. We don't need any intermediate host for it. Example is Giardia lamblia, Estamoeba histolytica. You understand? You don't require any intermediate host. You don't need any vector. You just contact it, maybe through kissing, through sexual intercourse, through contamination of water, through consumption of contaminated water, consumption of contaminated food, and so on. An indirect life cycle requires a what? Requires a a particular vector. It requires an intermediate host for what? For indirect life cycle. We now have the developmental stage of what? Of um of parasites. We have two. We have the homogenic development. All right, homogenic, and the second one is heterogenic. Heterogenic development. And the homogenic development is a developmental process where the generation of both parasites, where the generation is both parasitic and free living. Where the generation is what? Sorry, where the generation is either parasitic or free living. Sorry, is either parasitic or free living. You understand? And the heterogonic development is where the generation is both parasitic and free living. I get me now. So whenever the generation is both parasitic and free living, that is uh that is heterogenic, marked in red. That is heterogenic. But when it is only parasites or free living, mark in black, that is homogenic life cycle. You understand? Now, that is all on introduction to parasitology. I hope you are able to learn one or two things in this particular video, and in the next video we'll be talking about the uh, parasitic protozoan. That is the next one that we'll be talking about. Thank you for listening.